that. And so just want to, good morning, everyone, or good afternoon. And this is great to see everyone. Welcome to the e-seminar. This is the last e-seminar of the year because next month is difficult to schedule due to holidays. So we made this one special with the 3 speakers. And we will record the talk for viewing and Q&A for another session the evening for those in a different time zone. So that will make it a little bit more special for this e-seminar. And I'm confident we're going to meet next year in person, and welcome. I'm looking forward for that. So I'm V. Srivastava, immediate past president of the American Peptide Society, and joining you from Raleigh, North Carolina. As usual, Wendy and Lauren will be moderating the Q&A session and will lead into a virtual social hour after the presentation. So Wendy, do you want to add anything for the session number 2 before I go for the seminar?
Sure. Yeah, just to let folks know, as Fed mentioned, this is a special event this time. So we have this session at 11 a.m. Eastern, and then we have a second broadcast at 6 p.m. Eastern for people who couldn't join this session. And that's especially useful for our colleagues in Asia and Australia so that they can log in later on. So if you have people that you think would want to see this, but maybe couldn't come to this session, send them the link to the 6 p.m. session. And also, if you want to join a second time, why not? You're welcome to log in then as well.
Good. Thanks. Appreciate it. So we made, as I mentioned, we made this a closing e-seminar series special. And if you recall, we started the e-seminar in January of this year. And the talk was on recent work on chemical ligation for Phil Dawson from Scripps. And we are closing the series with a focus on peptide synthesis and characterization technology. So you can see the peptide synthesis is still a backbone. Can't get away with that, right? So we have 3 speakers today: Professor Fernando Albericio, John Collin of CEM, and Ted Z from UCLA as a young investigator. And just to mention that Professor Albericio, he's a modern day guru of peptide synthesis, doesn't need an introduction. And he will give a special note on the perspective of synthesis technology. And then it will be followed by the 2 speakers of the today. So Fernando is not only just professor, he's also entrepreneur and is a good friend of mine. He does not need introduction like mentioned before. Just to briefly, for those folks new on this seminar, and also the new to the society, welcome to the APA Society. He's at the University of KwaZulu-Natal and in Durban, South Africa, and he's also full professor at University of Barcelona Organic Chemistry Department. He's published more than 900 papers, more than 60 patents in file, so many books, and co-authored for 4+ books. And lately, he has been working on development of multi-component platform for preparation of peptide and bioconjugation. So he has a long list of achievements, and I would encourage you to go to LinkedIn profile, those who are new to the society or to this to the group. But with that, Fernando, is all yours.
Thank you very much, Beth, for the nice introduction. I am trying to share my screen, and I take in the opportunity to thank the American Peptide Society, Beth, Lauren, Wendy, for keeping peptide science alive during this COVID time, this terrible COVID time. It's a pleasure for me to join today. I am talking from the Pacific Coast in South America, in Chile, Valparaiso. And for me, it's 1 p.m. and it's a pleasure to me. Okay, this Wendy asked me to talk to do a introduction about opening perspective on the history and future of peptide science. And of course, this is my own perspective. This is the analysis of the last 5 years of new drugs accepted for the, let me put my pointer. Yeah, accepted by the FDA. And you can see that about 5% of the new drugs were peptides. Also in the ADCs, sometimes the linker is a peptide. But this 2021 year is excellent year because so far, okay, we are almost at the end of the year, but you have new 7 peptides accepted by the FDA as a new drug. This means that will be probably this year will be 15%. We are right now about 40 new drugs, 15% will be a peptide, an excellent harvest for this year. This is my personal perspective about some of the highlights. I am talking about the synthetic perspective. I am not talking about the medicinal perspective, the synthetic perspective. And of course, 1954, Vincent Vigneault published the first synthesis of oxytocin. This is a 9 amino acid peptide, took several years the synthesis of this peptide and by several cohorts. Of course, probably the most important highlight is in 1963 when Bruce Merrifield published the solid phase peptide synthesis. But also around the same time, a little bit north of New York, was an important highlight. Jim Waters, that founded the Waters Company, sent the roots for the liquid chromatography. 1970, also in Massachusetts, Lou Carpino developed the smoke protecting group. This was very important, but also was important a few years later in both in Europe. Eric Atherton and Walt Shepard start to work with the smoke ferbutyl in the solid phase peptide synthesis. And at the same time, Johannes Meyerhofer, Art Philax, Hoffman and Roche in a company, Wally Dano was there also, Wally, start also to use the smoke ferbutyl solid phase peptide synthesis. About purification, I say that Jean Rivière at Salk Institute, 1984, published a seminal paper about the potential of the reverse phase chromatography for purification of peptides in a large scale. And finally, in 2004, Mike and John Collins proposed the microwave peptide synthesizer. I will concentrate a little bit in this science and technology, the solid phase, the liquid chromatography, and the microwave peptide synthesizer. Of course, Bruce Merrifield was laureated with Nobel Prize in 1994, but the beginning was not so easy. This is an excellent paper, and I recommend you to read from Garland Marshall. I think Garland was the first PhD student in the Merrifield lab, than the beginning of the solid phase peptide synthesis. And this was some words in the reviewer of the paper on JCS that they called that the solid phase peptide synthesis was not chemistry at all, was a travesty. And the reviewer asked it to be suppressed by the community. Also, some of the colleagues say that the peptide was appear, disappear, and they talking about deletion sequence than failure sequence. At the beginning, also I remember my first European peptide symposium in 1980 in Helsinki in Denmark was a big discussions about the feasibility, the validity of the solid phase peptide synthesis. Right now, looks strange, but it was true. This is I think it's a nice picture. This was taken about 1994 in the Louis Carpino laboratory in Amherst, in Massachusetts. Was the last visit of Bruce Merrifield to Carpino's lab. Here was Emmanuel Faham from Egypt, Fernando Ferrer, and has a new company in California. Was in that moment was PhD student for Carpino. Again, about purification. Jim Waters, this visiting the laboratory of Bob Woodward in got Nobel Prize in 1965. This picture is about 1973, And Jim Waters prepared the first liquid chromatography for the separation of the intermediates of the B12 vitamin developed by Bob Woodbury. I want to call your attention. This is the laboratory of Bob Woodbury. This is the postdoctoral fellow, and Bob is smoking a cigarette. And here there is the fire extinguishers, and was smoking a cigarette in the same laboratory. And finally, everything about the microwave synthesizer started also in the East Coast. This is Mike Collins with two of his colleagues started in the 70s about the microwave. I used to call them the Collins revolution, because as at the beginning I mentioned, for Merrifield, they call the solid phase peptide synthesis travesty. And the big questions that some of our colleagues is, are we sure to cook our peptides in a microwave oven? Means we can call cook to Collins, also we can cook the chef. I would say more the chef than the cook. And the answer is very clear these days. The answer is yes. We are happy to cook our peptides, our growing peptides in the microwave. And why am I talking about revolution? I'm talking about revolution because the microwave changed our minds about the concept of peptide integrity. In the past, even the elongation of the peptide, we thought that was more a biomolecule. But the elongation of the protected peptide on the resin is not a biomolecule by itself. It's more an organic product. And we have to treat these organic products as organic products. In that case, we can heat them, and even we can use 90 Celsius degrees for the elongation of the peptide. I will not show any synthesis. It is the only synthesis I show. Only I will call your attention that it's a 34 amino acid peptide. You can use backbone protection, no problem. But I use, because this 34 amino acid peptide was only carried out, taking only 4 hours, 25 minutes, and 3 ml of solvent. This means that for each amino acids, we use about 22 ml of solvent. In that case, I will say and propose very often to the Mike and John Collins to change the color from blue to green, because it's an excellent tool from green chemistry. Means the microwave is, without a doubt, a disruptive technology for accelerating peptide research in a green context. And what about the future of peptide science? Of course, we are sure that we have new drugs. As mentioned, this year we have already 7 drugs. I know that people, Lily, Jordi Alsina is very close to have a new peptide-based drug. But probably the most interesting part of the science will be in immunotherapy. Vaccines. Manuel Quimpatarroyo in Colombia has ended the first peptide-based vaccine for the COVID-19, but also the personalized medicine. And the personalized medicine is changing the rules of the Food and Drug Administration and EMA agencies. So far, we need 10, 12, 15 years for a drug to reach the market. And right now, we need in a few weeks, 4 or 5 weeks, to have from the sequence of the peptide to the use of this peptide with the patients. And for that, I think the Liberty Pro that SEM is launching in the market will be key for this personalized medicine. Also, I think peptides will have a great importance for developing new linkers for bioconjugation and new materials. And the analysis from what is the future from a chemical point of view? For sure, we have to green the process. And in the definition of green chemistry for polar stars, I think we have to reduce the use and generation of problematic substance. And for that, again, I think the most important will be the purification. We have to be more efficient in the purification and in the consumption of solvents. And again, probably one of the tools for that will be the prodigy. Then in the next lecture, John Collins will talk about this project. And also, we have to improve the atom economy. We have to think that ethmoglycine, only you will use from the ethmoglycine the 20%. Means for in ethmoglycine, we have 37 atoms and only 7 are incorporated on the peptide sequence, only 20%. The best in atom economy is phenylalanine, then we use the 40%, but for glycine, only you use 20%. And I want to finish this short presentation paying tribute these people: Robert Bruce Merrifield, Jim Waters, and Mike and John Collins, for combining science and technology for the advancement of the knowledge. And with this is done, and thank you very much. This is in the 14 languages that we are talking in our South African lab. We're talking 14 languages from India, from Africa, Portuguese, Spanish, Catalan, of course, English. Thank you very much. It has been a pleasure to do this short opening of this e-seminar.
Thanks, Fernando. This is a great perspective, not only peptide synthesis, but the green chemistry and microwave driven. I have also seen peptides, some of those peptides are stable up to 90 degrees for 2 hours. And I have done those experiments. So yes, peptide can be cooked. So thank you again, and appreciate it. So we will hold on the Q&A at the end, as we mentioned to you. So the next presenter is John Collins. He is the Vice President of the Business Development at the CEM Corporation. He did a MBA from Kellogg's School of Management, Northwestern University, and the Master's in Material Science from Stanford University. John is a business person, but he's very smart in technical science, so don't take him lightly. Just want to make sure. He is a peptide scientist too. So John, he has been a CM for more than 14 years and focusing on developing and researching microwave-assisted application for solid phase peptide synthesis and nanotechnology, enzyme digestion, and protein hydrolysis, where you can all apply microwave as well. And as you all know, and you heard from Professor Albericio, that CEM is a leading provider for microwave instrument and leading towards a new chemistry. So John is going to talk about combining enhanced synthesis and purification strategy for peptide synthesis. So with that, John, it's all yours.
Thanks, Ved, and thanks, Fernando. Share my presentation. Well, thank you for the opportunity to speak and share some of the work from our laboratory. Today, as Ved was mentioning, I wanted to share some updated results and work we've been working on for SPPS and also some new purification methodology we've been working on. CEM, we're a private company based right outside of Charlotte, North Carolina. We develop processes and equipment for improving peptide synthesis, both at R&D scale and more recently at the manufacturing scale. Our history is centered around the use of microwave for peptide synthesis and really using microwave to drive reactions to completion and high purity in an efficient process. So both in terms of reducing the standard reactions in SPPS, the deprotection and coupling processes, but also really looking to employ it in a way that's efficient in terms of the use of reagents, the use of solvent, and expand this from a R&D approach to also where it can be used at larger scale. So we have systems now using microwave that can work up to 15 liters reaction vessel sizes. So you can produce up to kilogram batch sizes using the microwave technology. We've worked on a lot of peptides that are out in the market of commercial interest in the diabetes field, liraglutide and semaglutide, really honing in the microwave processes for these to really try to get the purity as good as possible for their use in GMP processes. And this has worked well. Microwave one of the traditional concerns has been epimerization of amino acid residues, but what we've been able to do is utilize microwave under optimized conditions and really minimize epimers of all of the standard amino acids. And that using these in large-scale batches, they compare favorably to recombinant batches. And this has involved the coupling chemistry, the side chain protecting groups used, and optimization of the use of the microwave, as well as the resin itself, importantly. What's remained, though, and an area we've recognized for a long time, but have seen as a bottleneck in an area that's a challenge is peptide purification. There's very subtle differences oftentimes between the peptide of interest and impurities, especially as you get longer. If there's single amino acid deletions, subtle side chain modifications like aspartamide formation. There's also challenges with hydrophobic sequences sticking to columns and their shape coming out of the columns. So what we have looked and others have used these, and which guided us, is to employing smaller particle size, longer column length, and also the use of increased temperature. Recently, we introduced a preparative HPLC system that utilizes elevated temperature. And what initially drew us to this was that all of our analytical methods used elevated temperature, and we wanted to find a way to match the two so that we could get the best separation. This system here employs columns with diameters up to 50 millimeter. And depending on your flow rates, it either uses just a column oven with a sample loop prior to the column in the oven, or at higher flow rates, typically about higher than 30 mils per minute, a mobile phase preheater that comes in so that the whole temperature and process is uniform from the top to the bottom column, and also from the outside fully to the inside of the column. With this, we use particle sizes down to 5 micron and column lengths at about up to 250 millimeter. And then employ a focus gradient calculator where we look at our samples when they come out of our analytical system, and it provides a guidance for expanded focus gradient around that time to run on the prep system. And this has simplified the workflow from our side, because typically what we would do is do our HPLC analysis at elevated temperature, then have to run some scouting runs at ambient temperature to develop a method for our prep HPLC. Now we can do temperature matching, and when we match the phases on our column, it makes it so that we can go straight from the analytical system for our QC and an analysis of the crude right to a developed method on the prep system. One market in particular that we thought this would be beneficial for is the personalized medicine market, where there's the need for a real high throughput manufacturing process where you can make a whole series of peptides and really avoid optimization of each peptide, optimization of a purification method at each step. So we thought this was a good place to start to evaluate the combinations of our microwave synthesis, our elevated cleavage process, and then the elevated preparative HPLC process. So this was a published paper that recently came out. It has a series of 20 neoantigens. We utilized our systems. With the microwave, we also utilized a combined one-pot coupling and deprotection batch process, where at the end of the coupling step, we actually don't drain the system. We just add a small amount of concentrated base and just proceed directly from the coupling step right into the deprotection. This is really nice because you can save energy by avoiding a second heating step. The temperature of your solvent, you're reusing it. It's already heated from the coupling process. And there's some inherent chemistry protection based on the solution phase reaction of your base you add, rapidly quenching your excess amino acid before any type of insertion can happen. So for these, these 20 peptides, we employed microwave SPPS, 5 equivalents, amino acid, DIC oxima using excess DIC to drive the activation and the formation of the OACL isoduria. And then we added 0.75 mils of a 25% pyrrolidine solution up at 110 degrees for 40 seconds, and then 2 washes after the deprotection step, and then a 30-minute cleavage. All of these peptides were then run on our Prodigy system, employing about 12 mg per mil is the concentration. We used a 21.2 millimeter column, 250 millimeter length with 5 micron particle size at 40 degrees C. And we were able to get all of the peptides made at first pass. You can see here sort of a scan across all 20. Some were very pure, some did have impurities, and some the impurities were close to the peak. Here's a few examples you can see here of a crude and then the purified run. Here's another example where you had some closer impurities present, as well as a third example. The sequences here, after we ran them through and purified them all, we were able to get a nice, good purity for all 20 of the sequences using this common focus gradient method, and pretty good recoveries here. So final purities in most cases were above 95%, good recovery and overall yield. So the whole synthesis was done in about 24 hours sequentially on a single Liberty Prime unit. The purification time was 10 hours, and per peptide was about 25 to 35 minutes. For many of these, we probably could have gotten away with a shorter length column, which would have reduced the purification time down further. We then have looked at, and there's been some nice work in the literature in the last few years on using flow synthesis at elevated temperature. We've obviously been using elevated temperatures through the microwave through a batch process. And in general, as the field has a need for longer and more complex peptides, as this is growing, the ability to make longer sequences getting into the protein realm is very beneficial for both for research and in general modifications. In this work that was done through the Pentalute group at MIT, there was a series of peptides made using chem matrix resin in a flow system with a very fast synthesis time of about 2.5 minutes per residue, with a flow rate of 40 mils per minute out of the column. And it involves a preheating of reagents before they're exposed to the resin, and in addition to the column itself being heated, with activation using HATU, PIOP, and temperatures around 90 degrees C. It's interesting when comparing batch versus flow SPPS, particularly at elevated temperature, and how the methods are optimized. With batch, you can obviously synthesize rapidly with the microwave. You have the ability to use elevated temperature, but it's in a batch process, it's more suited for carbodiamide-based activation because you have your reagents remaining on the resin for the whole time of the reaction. With the microwave, you can easily verify temperature and time conditions at any step within a run, just by modifying the amount of microwave that's applied at any step. You also have the ability through this to perform the one pot process, where you can keep your reagents remaining in there at the end of the coupling and add back to it a deep protection. It's also advantageous when you use greener, higher viscosity solvents, such as Tamasol, because with this one pot type process, you have less energy to flow through a filter. The volumes you're using and what you're pushing through the filter is less, which is helpful with high viscosity. With flow, you can have the ability to synthesize extremely rapidly, more rapidly than the approaches we've done to date with the microwave. There's been cycle times reported less than a minute, and for these proteins, less than 3 minutes. You also have a unique advantage where you can wash all surfaces of the reaction chamber by filling the whole volume with liquid. You naturally touch all the walls and the surfaces, and so anything that you'd have that go on the walls could get washed and flowed out. Interestingly, though, at the higher flow rates, the process appears more suited to onium salts versus carbodiamide process, because with carbodiamides, one of the rate limiting step is often the formation of the O-icyl urea, and in that case, you would want to have formation of your complete activated species before you start flowing across a resin. So otherwise, you'd be wasting amino acid that wouldn't be activated while it starts to touch the resin. Also, to minimize epimerization, because at these high temperatures with the bases employed, higher flow rates are useful for minimizing epimerization, so that it can get in there, but you can get it out before the activated species is epimerized. With that can lead to higher reagent usage, because you have a short residence time on the resin of any particular activated species. Realizing one of the disadvantages with the batch approach is the cleaning of the surfaces of the walls, we've recently started employing a headspace cleaning where we use nitrogen that comes in through the top, flows out, and as we're using higher temperatures for the base deprotection, we're above the boiling points of the bases often, whether it's piperidine or pyrrolidine. So you can get, and when you're bubbling too, you're inducing evaporation of the base. By purging nitrogen amounts into the vessel, you're basically forcing a high air exchange rate in the overhead space there, and that can remove and lessen condensation on the walls and within the surface of the vessel, which can be small amounts and can be subtle for shorter and medium length peptides. But when you get to longer proteins, these small amounts can add up, and this can be beneficial. So we wanted to see how a batch process could do for a number of the proteins that have been published in the literature with the flow process. So we utilized an optimized microwave batch process where we used a PEG polystyrene resin, or Peptide resin, 0.2 millimoles per gram. We loaded it at 0.05 millimoles scale using our standard 0.1 millimole volumes. We had a cycle time here. We employed typically a 4-minute coupling at 90 degrees C. So our average cycle time was about 7.2 minutes per residue. Used a combined 1-pot coupling deprotection process, and the overall equivalence is about 20 equivalents of amino acid. And then the deprotection was 1 minute at 110 degrees C, and then 3 washes of 4 mils after the deprotection step. We screened a number of peptides, and the first one was ubiquitin. In this case, about 76 amino acids. It's got a wide range of functions found ubiquitously in the body. In this case, we used a little bit shorter cycle time actually for the coupling. We were able to get the protein made in about 6.5 hours. We purified it then on a 21.2 millimeter column on the Prodigy at 40 degrees C, about a 25-minute method. And we're able to get a fairly good clean fractions coming off of the Prodigy. We also looked at, this was another example that has been demonstrated, a bar star a protein. I believe it's about 86 amino acids. In this case, we synthesized it using the microwave process in about 11.5 minutes. We then purified it at 40 degrees C with a 35-minute focus gradient. and then analyzed it on our UPLC-MS, and we're able to get what we believe to be a fairly pure fraction from the target. Proinsulin was another sequence we looked at, the precursor for insulin. This sequence can be challenging. It's got a number of cysteines in it as well. We were able to make this sequence on the microwave with a fairly good amount of target in the crude peak, and then we subjected it to purification. In this case, we used a 60-degree temperature, which we found was beneficial, and we're able to get some nice, relatively high purity fractions coming off of the prodigy using the same column, the 21.2 millimeter with the 5 micron particle size. HIV protease, we also attempted to make this sequence. It's 99 amino acids. We found this is also a very challenging sequence to make. In this case, we were able to identify the target in our crude product. We then subjected it to purification. Also 60 degrees C we found was the best temperature for this one in our purification. And we're able to get a nice fraction that's got a good amount of high purity target in it. There's also a collagen sequence we saw that had been demonstrated. In this case, it's 99 amino acids. We also were able to make this one. We did see some, in the mass spec, some cleavage of some of the backbone at the proline residues. But we were able to identify this as fragmentation and identify that the product is fairly clean here. 60 degrees again for this peptide seemed to be the best purity. We also looked at a little bit longer protein. This is a MDM2, 118 amino acids. It's got a his tag on the end, and it's involved in inhibiting P53 and has a number of functions that can be related to cancer and other things for research. This was definitely a difficult peptide to make. We were able to identify target. We did employ the use of an ASP gly at a DG segment here, which also brings the benefit of a backbone kink that can help with the synthesis. And we did also use 2 gly HMB residues that we put into the sequence at certain spots and double coupled the next residues and found that this was beneficial. Purification, this worked well at 60 degrees C, and we were able to get a target that we felt had a good amount of high purity protein in it. As a summary of using the batch process, we were able to make these sequences. We used 20 equivalents of amino acid. Comparing it to flow, it's a slower process than flow. It's about, in this case, on average, I'd say about 3 times slower. Its advantages, though, is that it uses significantly less amino acid, about 20 equivalents to, I think, around close to 100 equivalents for the flow process, and then the waste is about 4 to 5-fold less. Some of our future work with these is to try to work with these sequences and see how they would do in some biological assays, and also try to continue to work on the purification method to see how we can hone in on the purity even further. Is a technology summary, then I think what we feel that some of the advantages of these features are, is one is optimized synthesis conditions using microwave heating. You have access to automation, high purity, high efficiency synthesis. which can be useful for R&D, for neoantigen production, and is also scalable for peptide GMP production, and can really meet the specifications required for epimerization and impurities. Also, optimized chromatography. I think the elevated temperature really is a nice variable that's useful and can help the overall process. And with the Prodigy, it's applicable to peptide loadings up to about 1.5 grams onto the columns at 50 millimeter diameter. So at this point, I think another thing to add is I think the upper limit on stepwise synthesis is increasing, but purification is complex. And I think that'll be something that'll continue to have to be worked through and continue to optimize the synthesis conditions. But it's encouraging for the field that I think we're able to apply these methodologies and to access more proteins that can be made. Also, our technology now, we're working with Ambipharm to utilize this whole microwave process in under GMP conditions for peptide new chemical entities, peptide cosmetics, and we're excited to see the benefits that this can bring in terms of peptide purity and reagent usage, and ultimately lowering the cost of peptides to the market. And with that, I'd like to thank our team here at CEM, in particular Sandeep Singh, Drew Sesto, Travis White, Colin Simpson, and others that really drove this research, performed all the work in the laboratory, and are continuing to work to push and develop these methods, as well as our overall team here. Also want to thank Fernando, Albericio, and Beatrice on the great collaboration we've had with them on peptide synthesis and green chemistry, as well as Mike Pennington from Ambipharm, who we've had a lot of good chemistry collaboration with. And with that, I thank you, and I appreciate all of your time.
Hey, John, this is a great presentation, very clear, and also it shows the capability of microwave to the 127 amino acid synthesis. So it's coming along well, not only just green chemistry, but solving a lot of protein-protein interaction projects will be solved because synthesis for larger peptide is still a challenge faster. So this is great. Thanks a lot for presentation. And so with that, I'll move on to the last presentation from the young investigator. And John, again, thanks for Q&A will be at the end. So the next presentation is from young investigator, Ted Z. And he's, let me try to see if I Chih-Te, right? It's a short form Ted, no, or the longer form, the Ted. Anyway, so he's a PhD candidate in biophysics at UCLA. And Ted attended UC San Diego as undergraduate student, where he conducted research with Prof. Alex Grossman. And after graduation from UCSD, he moved on to Ferring Pharmaceuticals, and there he was for almost 3 years, working on peptide therapeutics and gaining experience in industry. And he decided to go back to academia and do PhD with Professor Jose Rodriguez, UCLA, and hope he is going to defend his thesis very soon. And the TED Talk, for 10-minute TED Talk, will be on developing methods in electron diffraction and Racemic crystallography. So with that, I will leave to you, Ted. All yours.
Thank you, Ved, for the kind introduction. And let me share my screen. And I want to thank APS for the opportunity to present my work as a 5th year trying to graduate very soon. This is a very good opportunity. So the primary question I want to answer today is why use electron diffraction? And the single most straightforward answer that I can give is that it really allows us access to previously unsolved structures by allowing us to look at crystals or diffract from crystals that are much smaller than traditional X-ray techniques will allow. So I want to highlight the differences between the two techniques. Here on the left, we have an X-ray diffraction pattern, and on the right, we have an electron diffraction pattern of the same proteins peptide sample. And the only difference really is the size of the crystal that was diffracted. So for X-ray, in order to collect a good data set, you really need crystals in the micron range, whereas for electron diffraction, you only need crystals that are in the sub-micron range. In fact, if the crystals are too large, you can't get transmission in the electron diffraction apparatus in order to even get a diffraction pattern. So what this causes is that the electron diffraction technique is actually a complementary method to X-ray. So in cases where you can have the luxury of growing crystals large enough to get crystals large enough for X-ray diffraction, I would do X-ray diffraction. But in a lot of cases, and I'm sure if anybody here has done crystallography before, sometimes it's just not possible to get crystals large enough, and that's where electron diffraction comes in. So just to give a very quick example of where it came in useful for our department. So in collaboration with the Steve Clark lab, and specifically with Dr. Warmack in the lead, we were able to solve what I think is still probably the longest peptide structure from electron diffraction 2 years ago in 2019, and this is of a truncated A beta segment, A beta 2234. And in this case, I just want to highlight here the crystal size. Sorry, the crystal size here, you can see that you can barely see it. The crystals are bunched up in a bottom of a 1 mil epichromatic tube. So, the next question I really want to answer is why, despite the fact that electron diffraction is a very useful technique, why electron diffraction hasn't been used more routinely, and why you don't really see that many structures being solved this way. Well, one of the primary reasons is that there is a dearth of phasing methods for electron diffraction. So phasing is the mathematical retrieval of phases from the diffraction patterns we collect, and that phase retrieval allows us to get the final 3D atomic structures. So when I joined the UCLA chemistry department in 2016, decades of beautiful work has already been done with phasing for X-ray diffraction data sets. So there's a plethora of techniques available for X-ray diffraction. On the other hand, for electron diffraction, there were only really two techniques available for phasing in electron diffraction. And these two were direct methods and molecular replacement. And I can't really get into details here, but both of these methods have weaknesses that prevent them from being used on a more routine basis for all samples that are interesting for peptide and protein chemists. So at the start of my PhD, Jose and I really wanted to hone in on racemic crystallography. And the reason why is that racemic crystallography really affords major advantages when you're looking at crystallography and phase. So this is beautifully summarized by Stephen Kent and Todd Yates in their 2012 review. It's a good read. I would honestly recommend anybody to read it, even if they're not a crystallographer. One of the first advantages that racemic crystallography really affords is the expansion of the space groups that you see. So here you have the observed space groups that are present in the PDB database in 2012. And if you, Todd did some calculations and predicted that if we started crystallizing stuff in there and racemically, you would expand the crystals that you would see by a lot, as you can see from this bar graph. So the racemic crystallography really lends to the ability for us to potentially grow more crystals and get more crystal bits from our crystal trials. Okay. The second reason why racemic crystallography is useful is because it really simplifies the phasing problem in many cases. So for each reflection in a diffraction pattern, there is an intensity and a phase value. So during data collection, the phase value is lost. And in a acentric crystal lattice, the phase value can range between anywhere from 0 to 360. And keep in mind that every single reflection will have a value, a phase value. However, when you introduce a mirror or glide plane into the mix, what you get is a mathematical restriction of the phase value to 0 and 180. So what this does is that it simplifies the mathematics behind the calculation immensely, where instead of having to figure out the phase between 0 to 360, now you're only limited to two choices of 0 to 180, 0 or 180. Okay, so knowing that, how does this effect help us phase? So although racemic crystallography is not necessarily a phasing method of its own, what it does is that it affords benefits to a lot of the other phasing methods that are used by X-ray crystallographers. So by utilizing racemic crystallography in conjunction with the other phasing methods, we were hoping as a group to translate as many X-ray diffraction phasing methods to electron diffraction phasing methods as possible. So this was basically what I started off on my first and second year of grad school. And here I started off with crystal trials of a peptide sample. And here it shows 96 well trays, which each well being a different screening condition. And we exposed both enantiomally pure, so L and D peptides to these conditions. So as you can see here now, that with the introduction of racemic crystallography, we are able to get a lot more crystal hits as indicated by the heat map. So obviously the mileage you get from racemic crystallography will vary sample to sample, but in our case, it really gave us a lot more crystal hits. So moving on, we were able to solve the crystal structures for the homochiral and the racemic crystals, and this is the racemic version of the crystal structure. So you can see that they're separated into L and D sheets. And most importantly for this paper, and in terms of our goals of translating as many methods from X-ray crystallography to electron crystallography, we were able to prove that essentially the simplification of phasing is preserved from X-ray to electron, despite the fact that in electron data collection, the pathway of the electrons from the source to the detector is much more convoluted by a variety of factors. So just to explain this figure a little bit more, when you in both of these cases, we released the artificial constraints that the software imposes on a centric data set by processing it in an acentric data set with equivalent symmetries, except for the mirror or glide plane space. And what we see is that despite the fact that we're releasing that restriction, the phases still bundle around the 0 and 180 degree region. So in terms of developing methods for electron diffraction, with that paper, we are able to show that racemic crystallography can be transported from the X-ray realm into the electron diffraction realm. And my follow-up work in my PhD has been to utilize that advantage to try to prove the other methods can also work in electron diffraction. And in parallel to that, other people in the lab, specifically Logan Richards and Maria Flores, in collaboration with Professor Usan from Barcelona, have proven that fragment-based phasing also works in electron diffraction. Okay, so with that, I would like to thank a lot of people. So first, the people in my lab, Jose, of course, as my PI, and Marcus and Callie, who have since become alumni of our lab, for their help with the Racemic paper in IUCRJ, Maria Flores for all her help with cryogenic techniques that our lab uses very often, the UCLA DOE for being one of the best run facilities, in my opinion, in UCLA. And unfortunately, I was unable to really talk about this collaboration as the data is still unpublished, but I am currently collaborating with Sam Gelman and Kate Corrigan, who has just graduated, and Isabel Wusan to really look at macromolecular structures that are seen as well. Okay, thank you.
Thanks, Ted. Great talk. Good to see electron diffractions versus X-ray. This is good. So I just wanted to thank all the speakers. Within an hour, we have covered from perspective on peptide synthesis and towards green technology, synthesis and purification using a microwave synthesis, flow, and also novel technique, and then also characterization of the peptide using electron diffraction. So from A to Z, we covered a lot in overview, mile high view. So thank you all the speakers, and I will leave to Wendy and Lauren now.